hold that carrying forward.
I don't know how Zach's gonna be walking in this.
You want to call it 100-yard touchdown, that's a 22.4.
But he did.
Here we go. Oh, I said
Anything like this, like an interest.
I've always felt so.
Yeah. I'm picking up.
Okay, so just a couple more things to say about skin, and then I can spend today's class period and maybe a couple more working through some of the other really important fundamental information about the anatomy of blood vessels and connection things in the nervous system and the anatomy of skeletal muscle, all introductory material that we will cover in much more detail as we get to those sections. So last time we had finished by bringing up the idea that the hair follicles are attached to the smooth muscle pieces in the skin. They're found running through the dermis and typically all the way up into the papillary layer of the dermis and then anchoring down near the root, the hair root, and when they contract, they tend to pull those hair follicles vertical. Now, when I introduced smooth muscle to you the first time, I told you that smooth muscle is found in hollow places. And now that's something we want to say again. And we're going to look at blood vessel anatomy here shortly. If you find a hollow place in the body, like blood vessels and lymphatic vessels, like the airway, like the digestive tract, like the urinary tracts, where you find these hollow places, you very much expect anatomically to see epithelial linings. Now, you should be able to identify many of those already. The next thing you should be able to identify in the walls of those hollow things is a layer of smooth muscle. And most of those places, in fact, all of the ones that I just listed, have a type of smooth muscle called single unit smooth muscle. And this is a cartoon diagram indicating to you the anatomy of that smooth muscle. This is not what the smooth muscle of the erector pili looks like. So these single unit smooth muscles have gap junctions between them. So they tend to behave as syncytiums, right? Single units. The same explanation that I gave for heart muscle. But look at the innervation. This is going to become important to us. The innervation here of the single unit smooth muscle, those sheets of smooth muscle that exist in those hollow places in the human body, are not innervated in the same way that the erector pili muscles are innervated, and they are not innervated in the way skeletal muscles are innervated. So these are three different innervation patterns to make note of here. The innervation of the single unit smooth muscle is innervated by what we call an autonomic varicosity. And you can see here that there is not specificity. The anatomical connection from the autonomic fibers on single unit smooth muscles do not have specific control of individual cells. Do you see that? Instead, the endings of the autonomic nerve endings have these swellings in them. Now, we're going to see that again, the swellings at the end of the projections. These swellings are where the chemicals are that stimulate the contraction of those muscles. And so how does the anatomy reveal to us the way single unit smooth muscle works in the body? It's a much more non-prescriptive, it's a much more non-specific kind of function. These things are supposed to contract in sheets. You don't want individual control here. So that's what the anatomy shows you. They just pour these things, they just pour this chemical onto the surface of the single unit muscle, and then it contracts as a unit, all the cells together, which is something you could have figured out anyway because of the gap junctions between the cells. So that's most of the smooth muscle. This is the way multi-unit smooth muscles contract, and they are located in very distinct locations. We're going to see them in the eyes when we get to the visual system, but this is where most of them are here. The multi-unit smooth muscles of the erector pili. And you can see they're different. Can you describe the differences in the anatomy of an erector pili versus the wall of a blood vessel? Now the cells are going to look similar. The spindle-shaped cells, they're thin on the edges and thick in the middle. That's typical, prototypical smooth muscle cells. And if you look inside the cell, they're not striated. So how would you describe the differences in the anatomy here? Looking at this picture.
No gap junctions.
No gap junctions. That's a critical observation. Okay, so what does that tell you about the function?
They would contract individually.
Okay, so this is going to lead to more specific control, choosing individual cells to contract. All right, last thing then, one more observation. Talk to me about the varicosities.
They're interplaced.
They're intertwined, yes. You see that? Intertwined varicosities in the multi-unit smooth muscles. Does that speak also to specificity? Sure it does. Now, let's compare this varicosity situation right here, to the skeletal muscle. Now, we're going to do this one a lot because this is the main focus of anatomy for the next 6 weeks, controlling skeletal muscle. So you can see here that there are individual nerve endings here on to the muscle cell. How is this arrangement, so this is a single muscle cell, how is this anatomical arrangement for skeletal muscle different from that one? Is this one more specific or less specific? Okay, so this is an individual multi-unit smooth muscle cell. Notice the number of varicosities, packages of chemical for stimulation. Notice how many of them are associated with that cell. See it? And what about this over here? What's the difference? What if I pointed out to you, would this help? What if I pointed out to you that the muscle cell membrane in skeletal muscles, everywhere the end of the neuron is that connects to it, has an indentation. Almost like the end of the neuron is plugged in to the muscle cell membrane. Now notice, it's not touching the muscle cell membrane, but it is very cell specific. Is it possible that this control mechanism here would stimulate other muscle cells? No. What about this one? You see that? Okay, so the varicosity really says non-specific. But for multi-unit, we have to say at least the anatomy suggests more specificity than the single unit arrangement. Okay, so this is the way erector pili muscles are controlled, and this we'll also see this again, like I said, when we get to the eyes. Talk about the ciliary muscles in the eyes and the control of the lens. Okay, now I want to remind you what this looks like. This was the section that we looked at before, so as you're going back reviewing this, I just want you to go back in your notes and see this. You don't have to do that now, but I just bring it up here as you're going through your notes. This is what we said originally, so I have added to that. That's the point here. I told you about the smooth muscle, I told you where it's located, and I said later I'm going to show you a multi-unit smooth muscle. Now I'm going to add one more piece to that today. So we're building our anatomy knowledge as we go. All right, let's finish the skin first before we do that. So skin cancer has to be mentioned. This is why you learn the layers of the epidermis. There are three major types of skin cancer: basal cell carcinoma, squamous cell carcinoma, and melanoma. Now let me open this with a story to get you thinking about it. Thinking about the tissue stuff that you've learned. My dad had a friend several years ago who was diagnosed with cancer. And the cancer was, they told him it was a squamous cell carcinoma, and it was in his airway. And my dad said to me, because I teach my family anatomy things, he said, "Isn't that skin cancer?" Now, how would you answer that question? He had a squamous cell carcinoma in his airway. Is that possible? Yeah. Right, so I just point this out to you. So once you learn the phenomenon of the epithelial linings, you see stuff like this, it's not confusing. Is squamous cell carcinoma a very common type of skin cancer? Yes. But could you make squamous cell carcinomas in other places? Yeah, that's normally what an esophageal cancer is as well. That's stratified squamous down there, where all that acid is pouring in on the esophagus and causes the unnatural mitotic rates there in the lining. Those are stratified squamous epithelial cells that are making those cancers. Okay, so just to get you interested in this, now we're going to identify just very briefly where these cancers are in the layers of the epidermis, and then a little bit about treatment, just to get you thinking about fun things in the skin. Okay, the basal cell carcinoma is the least malignant, so you got to cut it off. It grows very slowly and it metastasizes very slowly. It doesn't often do that. So this is the nature of a basal cell carcinoma. The question anatomically is, where are they? So if you've learned the layers of the epidermis, you know where this cancer is, right? We had corneum, granulosum, spinosum, and basale. Where are the basal cell carcinomas?
So,
In the highly mitotic cells of the basal layer of the epidermis. Is the epidermis, okay, first, how do cancers normally spread? How do cancer cells spread? Through the circulatory system, exactly. Is the epidermis vascularized? So where is the potential for metastasizing in the skin?
The derm.
In the dermis. Okay, so this cancer cell, the stratum basale basal cell carcinoma, is in the bottom layer. How does it spread? It's got to puncture through the basement membrane. And from what I just told you, is that likely? It's normally unlikely that these guys puncture through. It's the nature of these basal cell carcinomas. Okay, the second variety is the squamous cell carcinoma. Is a little bit more dangerous. These metastasize if you leave them, so they normally need to be cut off. They come up most of the time on your scalp, on your ears, and on your lips. But the prognosis is normally pretty good for squamous cell carcinomas. Where are they? They're in the stratum spinosum. Now, where are those cancers? Here. They're in the stratum spinosum, right? In this middle layer, if you will, of the epidermis. And so the prognosis is normally pretty good with those. Here's the last one, the most dangerous, melanomas. This is the most dangerous kind of skin cancer, and you might add to this, it is arguably one of the most dangerous types of cancer, period. A lot of young people are not aware of this. I'm quite certain that the young people that lay around in the sunshine without protection on their skin are not aware of this. The survival rates for diagnosed melanomas are some of the lowest of all of the diagnosed cancers that we know about. So they're extremely dangerous. The cells are highly metastatic. Where are they? Where is the melanoma?
in Germans.
Where? The stratum satellite, yeah. So we looked at that before. That's where your melanocytes are, down here in the bottom of the epidermis. So unlike their cousins, their cancer cousins, the basal cell carcinomas, these guys metastasize very quickly. They puncture through the basement membrane. They normally get into lymphatics. They spread in the circulatory system, and they cause all kinds of problems. And they're very resistant, as this says, to chemotherapy. So students want to know, how do I locate it? How do I tell it is a melanoma? Well, simple rules. ABCD rule for melanomas. It's asymmetrical. It has an irregular border. It has colors, and it has a diameter bigger than a pencil. Let me explain. What do you mean when you say it's asymmetrical? Okay, so most of the time, people are concerned about moles with melanomas. And moles are, in fact, local accumulations of melanin. Freckles are local accumulations of melanin. So when should we worry about them? Okay, so first, asymmetrical. Are most moles asymmetrical? No, they're little round, symmetrical objects. What about freckles? Freckles are oftentimes, not always, but oftentimes they are asymmetrical. All right, so let's go to B then. They have an irregular border. What's the difference in being asymmetrical and having an irregular border?
Different shape versus like the color changes as it goes around it.
Okay, so, yeah, you're right. I'm not to the color, you're a little ahead of me. I'm just trying I'm looking at a freckle and I'm looking at a mole. I can see that the freckle is asymmetrical. The question then is, does it have an irregular border? So when I say asymmetrical, I'm really thinking about the outer edges of whatever the structure is, freckle or mole. When I say an irregular border, I mean the top of the object that you're looking at. So if the mole is perfectly round, but it has crags and crevices in it, it should be watched. Follow me? Normally freckles are smooth on the surface of the skin. They don't have crags and crevices in them. Most freckles have no danger whatsoever. So it's asymmetrical and it has irregular border, and then colors. Certainly moles have a dark brown to black coloration on them. But if you see crags and crevices and streaks of reds or oranges in them, this is really high danger level. Different colorations in the mole is an indication that something bad is going on with the melanocytes. And then finally, the size tells you something. You always watch to see if there's changes in the size. And generally speaking, if it's bigger than a pencil eraser, you should go and have a professional look at it, probably cut it off. All right. So melanomas are treated with all kinds of fancy chemotherapies, immunotherapies. They're very dangerous, highly metastatic. And look at this. If the melanoma is more than 4 millimeters thick, then the chances of survival are very, very low. Why does that number 4 millimeters mean something?
That's it varying down into the dermis.
Yeah, right. So a typical section of skin at its thickest point is about 4 millimeters. So you can pretty much bank on it. If it's more than 4 millimeters thick, it is metastasizing. All right, last thing on the skin is burns, and most people are aware of this. I just want to teach you one phenomenon, the rule of nines here. But there are first and second and third degree burns. A first degree burn is just epidermal. Second degree burns include dermis. So if you get a sunburn and you have liquid-filled blisters on your skin, you have a second degree burn. That's what that is. A third degree burn includes the whole thickness of the skin. These are horribly nasty, really potentially life-threatening burns if they cover more than 10% of your body, which is not much. The burn area appears this gray-white, sometimes a cherry red color. It doesn't hurt at first, at least not in the area of the burn, because you cauterize and burn all the blood vessels and nerve endings, so there's no action potential, ability there to send sensor information. But the pain's coming as the tissue begins to repair. This word means swelling. So there's normally any initial swelling or pain, but like I said, that comes with a third-degree burn. So first and second and third, epidermal only, epidermis and dermis, and third degree is all the way through damaging nerve endings and blood vessels alike. Now, the rule of nines helps us to estimate the severity of burns, and most burn units will tell you these are your standard rules. If a quarter of the body has second-degree burns on it, it's considered critical. If more than 10% of the body has a third-degree burn, it's considered critical. Or if the third-degree burn is in a critical location. Face, hands, feet. Can you think of another critical place? I don't have it listed here, but I thought surely they know this. Where's another really critical place? So my hands and my feet are critical places. Thick skin, there's a lot of water balance things that can be a problem there. Face is obvious. Where else?
Chest.
Yeah. Here in the upper thoracic region, this is a critical place right here for third-degree burns. So students want to know, and you should know, how can you very quickly estimate how much of the body is covered with a third-degree burn, and that, in a clinical setting, is done using the rule of nines. So the body is split up into divisible parts of 9. So here they are, 4.5 on the front, 4.5 on the back. The head is 9%, but you already know that's critical if it's the face. The trunk is 18 on the front, 18 on the back. So a burn that would cover all the lower abdomen and pelvis would be about 9%. That's borderline critical right there. Just a burn right here would be considered critical if it's third degree. 4.5 on the front and backs of the arms. So one arm, third degree burn, that's critical. 9% on the front and backs of the legs. So just the front of one leg is very close to being, it's 9%, right on the edge of being a critical life-threatening burn. So memorize the percentages of nines in the different regions of the body. This is a good, very quick and easy way to estimate the critical nature of a second and third degree burn. All right, now, where we finished this a minute ago, I was talking about the differences in anatomy of smooth muscle and skeletal muscle. Now, I want to pause here for just a moment and let you know something. This is a good time just to pause for just a second. At this point, I am off the reservation in terms of following the textbook. So if you're looking in your book and reading, which I hope that you do, the images that you've seen, the pictures we went through with the histology, with the exclusion of the stratified cuboidal and columnar, which is very rare. We did all the others. We talked about where they're located and how you identify them and all that stuff. You can follow that right along in your book. In the skin, you'll notice I followed along almost exactly along with the order of the textbook. Okay, but at this point, I want to get you ready for the rest of the semester, especially the next major section of this course, which is the dissection of the human body. In my opinion, if a student doesn't have a big picture idea about bones and muscles and nerves and blood vessels, then the student cannot possibly think and remember what they're going to learn about the human dissection. So you won't get anatomy presented to you this way probably ever again in your life. But I've been doing it this way a very long time, and I've had a lot of feedback over the years from students who go off to professional and graduate programs who tell me the way I learned it there at Louisiana College did not cover nearly all the detail I had to learn when I got to where I was going. But you built a foundation for me so that I had perspective on everything that I learned. And so that's going to be what I'm going to do for the next several weeks. I'm going to try to give you perspective. We're not going to learn everything about anatomy, not anything close to it. But we are going to learn big picture ideas with enough detail to challenge you as we go through it. So the rest of, all I have to say now before test 1 is introductory anatomy things that will help us for the rest of the semester. So it's basically a look at muscle and nervous system and bones. That's basically what's about to happen. Just the base anatomy of these fundamental things. And you've learned some of it already. You guys know what the blood vessel anatomy looks like already. We've talked about bones already. So some of this will be reviewed, but it's my effort to try to create a backdrop for you that we won't have to go through again in an individual chapter, but you'll just have it readily available knowledge for you all the time as you learn. Okay. Yeah, so do the smooth muscle, remind them. So we're going to talk about three types of muscle, and you've seen all of them. But during this first volley, during the first round here, I'm going to emphasize smooth and skeletal. And I'm going to emphasize it to you in this way. Smooth muscle is under the control of the autonomic nervous system. It contracts involuntarily. So you say something like that, you have no context for that from me. So why don't I just teach you the basic fundamentals of the autonomic nervous system, and then you can think about that. All right? Skeletal muscle is under the control of the somatic nervous system. Now, I have been very intentional with you in teaching you an outline of a sensory pathway already. I use the skin receptors, the skin innervation stuff to do that. So in your mind, you should already have a general idea. If I'm on Miss Frizzle's bus and I get on a Pacinian corpuscle, I know how to get to the brain. I know which roads to take. I take the dorsal side to the spinal cord. From the spinal cord, I take the spinothalamic pathway from the Pacinian corpuscle, probably on the anterior side of the cord, and I make my way all the way up into the brain to a structure called the thalamus. That's a long road, especially if the Pacinian corpuscle is in your leg. It's going to start low on your spinal cord, and that roadway goes all the way up to the thalamus. We call that pathway the spinothalamic pathway. Does that ring a bell? Okay, so I'm going to turn this around now. As we go through this, talking about these pieces, and I'm going to go to a muscle. So I'm going to start in the brain now and build another outline, this time from the brain to the muscle. And this will allow me to introduce to you all kinds of general principles that we find in the nervous system and how the nervous system regulates muscle contraction. Okay, so start with a little story to help them think about it. You know this anatomy already. So we're talking about smooth muscle. We're talking about smooth muscle information here. I stuck these slides in for review. Let me come back to this. Does that look familiar then? I just said it. I figured I was going to say it, so I just showed it to them again. Y'all remember this? There's the pathway from the Pacinian corpuscle to the dorsal, up through the various structures, synapsing in the thalamus here. This is the spinothalamic pathway, a sensory pathway. And I showed you for this deep pressure stuff that a lot of this information is on the front side. And these are highways, right? These are white matter areas. And what do we find in the white matter? What is white matter exactly? Axons. And what gives it the white appearance? The myelin, yeah, the insulation on the axon gives that appearance. So, like I said, as we begin to progress through this, you'll learn where all these are. So this is just your first one to get you warmed up to it. All right, so back to the blood vessel here. You know this anatomy already of this blood vessel. You know, for example, that the inside linings of the blood vessel are what kind of tissue? I call it the endothelium, or the tunica interna. But what is it anatomically? Good. So you all should have said that in unison. Simple squamous epithelium. The endothelium is simple squamous. You'll learn this, yes? This is a hollow structure, so what would you expect to find next? Yeah, smooth muscle. Now, notice something that I haven't pointed out yet. The tunica media on the smooth muscle layer of the blood vessel is different in the arterial system than it is in the veins. Can you see that?
Say that again, one more time.
The smooth muscle layer in the artery system is different than it is in the venous system. The anatomy is different. The smooth muscle. So answer me this, is this single unit or multi-unit? These are single unit smooth muscle sheets connected by gap junctions, innervated by varicosities. Okay? Now, what is the difference in the smooth muscle lining of the artery versus the vein?
The arteries have to be larger because they have a higher pressure going through them.
Okay? Well, they're not all larger.
Mainly bigger.
Arteries are not all larger. There are large arteries and large veins.
I mean like the circumference of, like the amount of muscle in arteries, there's more muscle in arteries.
Okay, now there's the observation. There's your observation right there, yes. The wall has more smooth muscle in it. And you're dead on, you're spot on with your observation about function. So arteries are oftentimes referred to as the resistance vessels in the body, where veins are referred to as the capacitance vessels. Arteries have the potential to resist flow. So if you're going to use the autonomic nervous system to regulate blood pressure, where will that regulation happen? The artery or the vein? In the artery, yeah. So if you want to design a drug to help with blood pressure, would you want that drug to make smooth muscle contract or relax?
Relax.
Okay, I got both.
It
Somebody want to
depends
give
on
me
what
a
we're trying to do.
why?
Are we trying to, we're
Depends
trying to
if we
lower
want
the
I want to lower blood
right?
pressure with a blood pressure medicine.
And relax.
I want it to relax. I want the lumen to be bigger, right? So there's less pressure in the system. And that's where, okay, now, it's sometimes shocking students to learn that the pressure in the arterial system is normally around 100 millimeters of mercury. So if you test blood pressure, normal pressure is around 120 over 80, systolic over diastolic. What do you think the pressure is in the venous system? It's got to be less than that, right? How much less do you think? 50? 20?
75.
10? 75? Anybody?
I feel like it's drastic.
You do?
I think so.
Why would you think that? Why would you think the pressure dip would be so drastic?
Because the differences in the veins and the arteries are really
Okay. Okay, yeah, so there's a dramatic difference in the anatomy. And you happen to be right about this. The average pressure in the venous system is less than 2 millimeters mercury pressure, which means that the veins have to have all kinds of special anatomical adaptations about them in order to facilitate the movement of blood through them. For example, most medium-sized veins have valves in them, so the blood's not allowed to go backwards. And most veins sit wedged between skeletal muscle fibers.
It's really a squeeze.
If you want to find a vein in your arm that's not superficial, so you have superficial veins in your arms and legs that you don't have arteries for, but if you want to find a vein in the body, you look in the muscle.